All right, we're live.
Thank you. Hey, Malcolm. Can I talk with you for just probably 2 minutes? Because
I'm just testing here to see if it's.
okay. Oh, yes, I am. You too. Very unfortunate thing. I just want to share with you because.
Look up here because you really are.
Oh. Oh.
If Rusty comes by, I put my information that he needs on the laptop. I've logged in and I put the information that he'll need to finish the process on that laptop. So you can just let him in and let him finish up. All right.
All right, now I want Brian, so I'm going to do the best too. I can act like a
Sure.
We are live right now.
I'm sorry?
We're live right now.
Oh, okay.
Oh, kind of started. How's that?
Yeah, that looks good. I think they're on this side, welcome. Right here.
Yeah, I'm just gonna do it. At least you said you would, so I got Okay, we've got plenty of time.
We are live on YouTube right now.
All right.
So just, yeah. Just be aware, just be aware.
Caitlin. Thank you. Hi. Oh, snap.
We gotta turn it around. We got lost in the elevator. We did. Where I saw her go in the elevator.
Yeah,
Do
it's
you
good.
want to put it away from her, because if somebody comes in late, they can go.
Yeah. Dang, guys.
Bags
on.
Yes, I put the whole bag in the fridge.
Okay.
So, yeah.
And that's fine.
Yeah.
Probably one page
Yeah.
Yeah, I was like, I have hips and I almost can't fit. I have that.
Alright, let's move this.
I'm just gonna get the book back.
Okay, you have the book back. What are you talking about?
I didn't know how this worked. And then I looked at your-
Sorry, I just saw you. You are just feeling better, but something's still going on. Someplace not going better.
I was like, as an asthmatic person, please. Don't go in the hospital.
I would prefer not to, yes. The goal is not to hurt anybody during my dissertation.
Alright,
Will do.
you know what? I think we need more cups though.
No.
No? Okay.
Okay, yes, ma'am.
So we pull the cup.
Yes, ma'am. Sorry.
I don't think a lot of people are gonna be here, so please help yourselves as much.
We did everything with gloves on and disinfectant hands underneath the gloves.
Listen, you had the
Virology.
virologist on
Yeah, so
supervision,
this
so
is
we're good.
totally safe.
Okay.
I know, Dr. Shark saw me walking on the hallway with lava sick. It's for food. There's no ethereum for my people. It's not allowed.
Oh, you guys are awesome.
Alright. Weird. Alright. What's that?
Hey, y'all, you know about bring your couches?
Oh,
Boss, you
Yes,
forgot
ma'am.
the small plates, the paper plates to put stuff on it. Good thing I'm always a glutton because I find that things like things back. Everything works.
There's the only grab them in.
So they can throw their
Oh, yeah.
thing in the.
Hey, Dave.
We'll put it over there.
You're Rada.
She's getting nervous, she needs a bucket.
Oh, online YouTube.
none of that, it's actually in frame.
You know, on your door.
It is live already.
Oh my gosh.
There's a case, this right after. Figure out the one.
No, I'm sick. I don't feel comfortable seeing a newborn.
Yeah.
With a potentially,
Yeah.
compromised immune system. Is this for me, right?
Yeah, all of this.
I bought a special t-shirt just to you today. It says, don't let the breathe it out. You need a blanket.
Great.
You look from there.
Rada, can you hear me?
That's Kathy. Hi, Kathy.
Hello, you look great.
I'm ready to go, yes. It's great to see you.
Good to see you. Did you get hold of Martina?
Yes, she will be joining us shortly.
Excellent. Okay, good. Okay, so it's all set, and we'll just be ready and, yeah.
Great.
You're all ready. I'm gonna call for the minutes. You just want to be over with.
Yes,
This is so good, you're gonna feel 3 hours.
yes. That's exactly what I'm thinking right now.
That's the way to go.
Yeah, so hopefully we'll all be good. But great, I'm glad that you joined.
Get ready and,
All right,
Okay.
will do.
sounds good. All right.
It's a great idea.
Oh, I know. He's good. Oh, yeah. His heart is going real slow. Yeah. Well, the babies. I don't know. Yeah.
I'm gonna find clothes.
And then I did not. No, I did not. This was an Emily had so Sean had almost like a warm for an a flat hair. And Emily, she always has short hair and it will not grow for the longest time. And her like a picture of 1 year old that she just started on. Now her hair is down here and it took us 6 years to grow. I mean 5 years to grow that long.
Right.
Yeah. But it's a lot.
Yeah.
It's gonna start popping off.
Right.
And growing. It's so cute. Yes.
Just driving past it, and I was like, that was like, nobody in that room. Just drove from home. Okay. Yeah.
I know you can't.
You remember Tracy? Yeah. I was gonna say it's been a while, but it's 30 years.
You're welcome. I'm so proud of you. I love you so much.
Thank you. This is pretty special.
Traveling.
I know.
But that's fun.
Something happens now, Jane.
Okay.
It was funny, I was talking to him on his boss, and I was like, I don't know what I do, but I pretend like
How have you been? I was in California. I was
None of that will be happening.
So Rada doesn't know what we're doing.
Yeah. Can you sign the card for me, please? It's so funny, Angie does this all the time. She grabs a notebook and she's like, pretend like we don't know what you're doing.
That is great, nice. He is able to. Oh, I have so many friends. At the age of 3, they still have economic it's long.
No, they love each other. There's not really.
I don't need the time now because.
I timed myself yesterday, it was 46 minutes. So I'm guessing it'll be a little bit shorter because, I'm a little more nervous now.
5 minutes already.
No, you're totally fine.
I don't want, to feel like.
It's totally fine. If anybody that needs to leave, you're welcome to. It's not gonna disrupt my
We
flow.
have that meeting that we go upstairs.
Yeah. No worries.
5 minutes.
No worries.
Stops to snacks and beverages that are over here, please. There's a lot of food. Please stand. There's coffee, too.
It's so funny how people react. It's so on my phone, it's everywhere. I saw y'all can go out and eat, but then I still go out and eat, people having your food.
Hey! Oh my gosh, how are you? Good. Thank you. I'm trying like this. Oh, yeah, that definitely is nice. Oh that's right. Super exciting. Yeah. Let's just do
Yeah.
So that's what it is. So they give you a really comprehensive first part, basically.
I don't think so, never. Don't recognize your face, I'm sorry.
Yeah, when it comes to analysis, it's one person. So far, yeah, you first.
Yeah. Oh my God. Sorry. Yeah, I'm sorry.
Caitlin said she was watching. Caitlin
I'll eventually just follow that.
And then things happening. Some type of information. I'm warning you. I got some kind of
No, they just, they have I actually, yeah, I got half of them. Come back for a while.
well, like yeah, I see her too. I just can't hear her. Her line is muted. There's Martina.
Good.
Yes?
Can you hear me?
Yes, can hear you.
Okay, so. Hi. How are you? I'm good. Yeah, right. Okay, last three. You're 19, I have to say that. It's good. Yeah. You guys closed? Yeah. Hopefully for like 2 weeks. And we, yeah, he got the basket.
Yeah, we're missing Dr. Wasserberg.
Yeah, then let's wait a little bit longer. We're missing one committee member here.
He's online. Sean.
Okay.
Sorry.
How are you?
Just want to say hello.
We have everyone.
Okay, everyone, I think we're going to go ahead and start. I'm here on behalf of Dr. Rupel to introduce my friend Rada Petric for dissertation defense. Rada joined UNCG for undergrad many years ago, and then she finished her master here as well. Since she was an undergrad, she already started working with Martina on rodent acoustic communications, and then she did her master thesis on that topic as well. After that, she worked here as a lecturer for 4 years. After 4 years, she decided to return to pursue her PhD with us. So let's welcome Rada. And now that I would like to highlight our committee members. We have 2 committee members. Dr. Martina Rupel is joining us through video, as well as Dr. Kathy Marler from University of Wisconsin-Madison, also joining us online. And here we have 2 members, Dr. Ramzi Bhandari and Dr. Gideon Wasberg. So without further ado, Rada, please.
Thank you. Right, so I would like to start off by acknowledging everyone who's helped me with this project, and specifically my committee members, my academic advisor, Dr. Martina Rupel,
and Dr. Catherine Marler, Dr. Gideon Wasserberg, and Dr. Ramji Bandari. I would also like to thank the people who helped me in the field. So I collected almost 3 years worth of data in the field site, and all of these people really helped me. And then I had a lot of undergraduate assistants who helped me sift through over 1 million audio files looking for mouse vocalizations. And of course, the research sites, funding agency, and logistics were actually making it possible for me to do field research. And I could not have done this without the support and unwavering love from my friends and family. So with that, I'm going to tell you about my general outline. I'm going to talk about acoustic communication as my background information. And my dissertation is divided into part is anthropogenic noise that has been before. I'm going to end my talk with a conclusion and future work. So vocal communication is an integral component of animal behavior. Senders' vocalizations contain information and elicit a predictable behavioral response in the receiver. Animals rely on vocal signals to mediate a myriad of daily activities, such as prey detection, predator avoidance, and mate attraction. And these signals contain information about species, sex, individual identity, and reproductive condition. And this variation is based on spectral and temporal characteristics, such as duration, frequency, and bandwidth. And I'm going to talk about these in a moment. But I want to mention that throughout this presentation, I'm going to show you several of these spectrograms that are going to have frequency on the y-axis and time on the x-axis. So in the top spectrogram represents 3 bat individuals from the same species. So here's individual 1, 2, and 3. And you can see that there are differences in duration of their calls. So bat number 1 gives a shorter calls than bat number 2. And in the second spectrogram represents 3 sympatric species.
Just gonna adjust because the computer doesn't really show the screen and the
Oh, okay.
background is too dark, so.
No worries. Okay, so 3 sympatric species of bats, and you see that they're vocalizing at a different frequency. So this is species number 1, and they're about 50 kilohertz, whereas species number 2 is much higher. Then we also have bandwidth differences. So bandwidth means the number of frequencies that a call passes through. So in bat number 2, or species number 2, there's a larger bandwidth than in species number 1. Okay. Does it come through? Is that all right?
I'm gonna try to move a little forward.
Okay. All right, so vocal communication is arbitrarily divided into 3 parts based on what humans can hear. And what I'm really interested in is the soundscape that's above human hearing range. And that's using ultrasonic vocalizations, are bats and rodents. And I'm specifically interested in rodents. So they make up almost 50% of the extant mammalian diversity. And both males and females regularly produce ultrasonic vocalizations in a variety of social contexts. So this image is really complex, but that's because the environment is really complex. So what we have here is 2 individuals that are interacting with one another via ultrasonic vocalizations, or USVs. And in nature, while these 2 individuals are interacting, there are a whole lot of other things that are going on in different species. And here's just an example of a bat call. And other factors such as wind and rain also influence this interaction. But at the same time, while all of these environmental factors are influencing the interaction, there are a number of different physiological factors that are influencing it simultaneously, such as age, sex, and reproductive condition. So vocal communication and other behaviors is sensory systems collect information from the environment, and they forward the message to the central nervous system for processing, which triggers a physiological response that alters cellular processes and induces a behavioral response. And for my dissertation, what I was really interested in is how both a physiological stimulus as well as an environmental stimulus influences both pieces in free living animals. So this leads me to the first part of my talk, and that's focusing on testosterone pulses. So for specifically looking at the physiological stimulus, what I was interested in is the androgen hormone testosterone. So in various animal species, behavioral endocrinology studies have demonstrated that testosterone is important for territorial behaviors, sexual and social behaviors, including vocal communication. And most of the studies that have examined the relationship between testosterone and behavior have mainly focused on these long-term changes in baseline testosterone levels. So the A and B represents baseline testosterone levels, and they're occurring over a longer period of time. But for my dissertation, what I was interested in is these transient increases in testosterone, or T pulses, that occur after social interactions. So I want you to know that these T pulses are occurring on a much smaller time scale, in minutes, as opposed to weeks or months. And T increases occur in various taxonomic groups, including humans. And the proposed function of these T pulses is to rapidly modulate male behaviors in a changing social environment. So looking at these two graphs, this is from a review paper. In rodents, testosterone pulses occur after male-male aggressive encounters and male-female sexual encounters. So time point 0 represents when the encounter occurred, and approximately 45 minutes later, you can see a spike in testosterone. So what is the proposed function of testosterone pulses? Well, T pulses have rewarding properties. They can condition animals to the physical location in which the hormone release occurred. So I want to talk about the monogamous California mouse. What's really interesting about these mice is that the effects of testosterone pulses on forming a condition-place preference is really context dependent. So what's really neat is that in pair-bonded males, but not unpaired males, so pair-bonded males form condition-place preferences for their home box or chamber, whereas they do not form it for the unfamiliar chamber. And this is a study that was done in the laboratory, where we see the opposite effect in unpaired males, suggesting that context is really important. But is this a lab artifact, or does this also occur in nature? Testosterone pulses can also have short-term effects or rapid effects. So I want to talk about California mice specifically and the rapid effects of these testosterone pulses on their call production. So California mice produce several different types of vocalizations. The SVs are the most commonly recorded, and they're produced by both males and females. And in the field setting, they're produced when a member of a pair is away from the nest or away from each other, suggesting that there's some type of a contact call. They produce simple and complex sweeps associated with sexual behaviors, and then barks are associated with aggressive behaviors. So in the laboratory, a single T pulse 20 minutes after the administration changed the number of vocalizations that were produced in paired males in response to a novel female. So paired males reduce the number of vocalizations that are associated with those sexual behaviors in response to a novel female, where we see that in unpaired males, it increased the number of calls, right? But testosterone pulses can also have long-term or greater than 24-hour effect on call production. So after multiple T-pulse injections, 2 days after those injections, and lastly, T-pulses can modify call characteristics. So in plainfin midshipman fish, within 5 minutes of a T-pulse administration, the actual male produced calls that were longer in duration and at a much higher rate than those males that were injected with either saline control or the non-injected females. So, but we don't know if T-pulses actually have these rapid effects and long-term effects in on call characteristics in nature. So manipulations of T-pulses have mainly been conducted under controlled laboratory conditions. And here I'm asking the effects of T-pulses in a complex field setting. And because what influences animals' behavior in a natural setting might be really different based on the competing stimuli that I introduced you to at the very beginning of the talk. And I want to note that there's a reason why this question hasn't really been answered in the field, is because it's difficult to study animals that are active at night, which we can't see, that are vocalizing in a range which we can't hear. So for my two objectives for this particular part is to look at the effects of testosterone pulses and how they alter spatial and vocal behaviors at two different locations. And the reason I selected these two locations is because social interactions at a nest site are going to be different from the social interactions we observe at territory boundary. So context, again, is really important, and it could alter individual behaviors or the responses of individuals to T-pulses. So to address my aims, I studied the monogamous California mouse because they form these long-term social bonds, and males must balance territory, paternal care, and mate attendance all at the same time. And the testosterone is an important mediator of all of these social behaviors. And they regularly produce these ultrasonic vocalizations that I introduced you to at the beginning of the talk. And I want to mention that in the field setting, they produce a lot of these SV calls. This is the most common type of calls that are produced in the field setting. And this is just what their habitat looks like. This is all poison oak. So live oak trees and poison oak. And so I studied these mice for 2 years in Central California, trying to answer my aims. And I want to note that these particular SV calls are produced in a sequence that range anywhere from 1 call all the way to 11 calls. And based on the number of calls in the sequence is how we refer to that specific call type. So this is 2 calls, that's why it's 2 SV. All right, so I used a set of general methods to measure behaviors of these mice. And what I specifically did, I put tiny little collars around the necks of the mice, and this allowed me to track their activity, figure out where their nest sites were, where their territory boundary is, and how much time they're spending in any particular area. So I was a stalker of mice. I also set up microphones to record their vocalizations. And I want to play this vocalization for you. This is a typical 2 SV call, and it's slowed down to 4% of its original speed so that we can actually hear it. And then lastly, I set up a thermal imaging camera to give me a visual of their activity in an area of interest. So what you're seeing here is a screenshot of the actual thermal imaging footage. And specifically, this is a nest site right here. And on top of it, the square is an antenna. That's basically what's collecting information about how much time the mice are spending there. And then this little white dot is a mouse. And so when a vocalization is produced, I was able to assign context, whether this mouse was alone or if there was another mouse present, and if that other mouse was close or far away. So for my aim 1, I looked at the effects of these testosterone pulses at the nest site and looked for specifically for spatial preference and changes in their vocalizations. So what I expected is that testosterone pulses would have an effect on an increased spatial preference because of that condition-place preference hypothesis. I also expected that mice would increase their call production and that their call characteristics would be different. And specifically, because they were at the nest site and these SV calls are important or most likely serve as contact calls, I expected they would increase these SV type calls.
So specifically, the way that I actually tested this is I looked at and identified paired mice. So remember, they had radio collars on, and if the signal from both the male and the female were coming from the same nest, I figured out that they were mated. But it also allowed me to figure out if they had pups at the nest or if they were lone parents. Okay. And so once at the nest site, I set up, this is a typical nest site, and I set up all of these traps to actually catch the male so I can administer the injections. And what I was able to do is I administered 3 subcutaneous injections over a 5-day period. And the males were randomly assigned to either receive a testosterone or saline control. And after the 3rd and final injection, I recorded their vocalizations for 3 consecutive nights. I recorded these behaviors from both the male and the female. So I recorded the amount of time they spent at the nest, number of and type of calls that they were producing, and call characteristics. Right. And so what I found is that testosterone males spend more time at the nest. And here we have our testosterone males and control males. And this is particularly exciting because these 3 subcutaneous injections but I also found that testosterone males who had pups at the nest spent the most time at the nest. So there's also an influence on pups based and injection to seeing how much time that they're spending there, with testosterone males with pups spending the most time at the nest site. I want to note that I did not inject the females, and I'm only using the name T females based on the treatment that her mate received. So T females spend less time at the nest site than the control females, which is really interesting because in this natural setting, it seems that the male and the female are coordinating their behaviors, and the female is responding to the altered time allocated at the nest site of her mate, and then she is moving away from the nest site. And again, pups were important in the female also. With testosterone female that did not have pups spent the least amount of time at the nest site. All right. So in summary, testosterone injections, which mimic naturally occurring testosterone pulses that are expressed after these social interactions, the male-male and male-female interactions, can increase the amount of time that a male spends at the nest, with testosterone males spending more time at the nest than the control males, which supports my condition-place preference hypothesis. And as mentioned, this is really exciting because this is occurring in a complex field setting. And the other part is that these T-induced CPPs at the nest or condition-place preferences could also be the mechanism for maintaining paternal care. So in California mice, paternal care is actually essential for offspring survival. And testosterone is positively correlated for paternal care in this particular species. And sexual encounters in this species are occurring at the nest site. So the males, so T-induced condition-place preferences at the nest site could be the mechanism for keeping the male at that particular site. So I find this fascinating, again, because the allocation of time and energy spent by the male at the nest site is altering the spatial preference of the female. So what is the female doing then? Well, if they're coordinating their behaviors, then the female is most likely either defending the territory and or maybe at the same time replenishing her energy reserves, right? And for the actual acoustic component, I found that testosterone pairs produce more of those, more SVs, so the sustained vocalizations, but overall they produce more ultrasonic vocalizations. And this is consistent with previous literature that testosterone pulses can alter the number of vocalizations produced. Okay. All right. I also found that T mice produce more SVs when they were alone. So they produce proportionately more calls when they were alone versus if there was another mouse, whether they were less than a meter apart, less than or greater than a meter apart. All right.
And finally, I found that T males produce lower bandwidth calls, which is not what I expected. So typically, when we think about testosterone, males produce more modulated calls, meaning their bandwidth calls are higher instead of lower. So testosterone has a positive effect, typically, on bandwidth. And this is the absolute opposite of what we see. But it's really important to start thinking about what is the functional significance of these calls at the nest site. So if the males are producing these calls at the nest, what is the function of it? So I want to talk about this a little bit more while we have the whole big picture in here. So the T pulses are altering the amount of time and social behaviors, and smaller bandwidth calls at the nest. So overall, this suggests that these could be a contact call to get the female to come back to the nest. And specifically, these lower bandwidth calls, as opposed to the higher bandwidth calls, they travel further in an environment as opposed to the higher bandwidth calls. All right, so in conclusion, testosterone males spend more time at the nest via condition-placed preferences, and they're not spending time towards mate guarding or courting, and most likely. And this is observed because they're not following the female. The female is spending more time away, and the male is spending more time at the nest site. If these behaviors were true, we would expect the male to follow the female. And they're also most likely not defending the territory because, again, they're spending more time at the nest site. Which brings me to my aim two, looking at the effects of testosterone at the territory boundary on spatial preference and vocalizations. So T pulses at the territory boundary, I expect that would have that similar effect where they would increase spatial preference based on condition-placed preferences. They would increase call production, but I expect that they would produce more of these bark calls because if you remember, they're associated with those aggressive behaviors. And then I also expect that the testosterone would change call characteristics. So the same methods were actually used as for the nest site as for the territory site. The only difference is that injected the male at that particular location instead of the nest site. So again, they received 3 injections of either testosterone or saline control over a 5-day period. Then after the 3rd and final day, I recorded the amount of time that they spent there, number of type of calls produced, and those call characteristics. And what I found is that both males and females spent less time at the territory boundary than the control males and the control females, which is not what I expected. So I expected to see that the males would spend more time, I expected they would spend more time at the territory boundary because of those condition-placed preferences, but that was not true for this particular case. So it made me wonder, what are these males doing? Where are they allocating some of their time? And one thing that I was able to measure was distance that they were traveling outside of their original territory. And so what's interesting is that testosterone males traveled much further than the control males outside of their original territory, which to me would suggest that it's promoting territory expansion. And finally, I did not find a difference in the number of calls produced between the control and treatments, but I did find a difference in the actual duration of calls. So testosterone males produced shorter calls than control males. And this is consistent with laboratory research in California mice that shorter calls are associated with aggressive behaviors. So they're most likely engaging in some type of aggressive interaction and producing these shorter duration calls. So in summary, my testosterone pulses at the territory boundary do not support my condition-placed preference hypothesis, but the T pulses are altering their space use. So specifically, testosterone males and females are spending less time at the territory boundary than the control males and females. And most likely what are the males doing? And the males are most likely challenging other males outside of their territory and engaging in those aggressive behaviors. But it also makes me wonder what the females are doing. And so if the females are spending less time at this territory boundary, then they're either going back to the nest site, they're actually defending other parts of the territory, or they're coordinating their behaviors with the male to actually secure more resources together. But all of this remains to be tested. So variation in testosterone pulses in inducing condition-placed preferences at the nest site, but not at the territory boundary, shows that there's plasticity in sensitivity to these testosterone pulses. And the environment is really important when considering this. And that is what I just said here. Should have clicked a little bit sooner. All right, so in summary for aims 1 and 2, in nature, testosterone pulses are able to alter time management and vocal behaviors. And again, this is really exciting because these testosterone pulses are changing behaviors in a natural, complex setting. And responses are highly context dependent and found in both sexes. So we see that the response at the nest site is different from the response that we observe at the territory boundary. And testosterone pulses are important physiological stimulus that can control the spatial preference and vocalization plasticity. And this is leading on to the next section of my talk. So how are California mice deciding to allocate their time and coordinate their division of labor? And they form pair bonds, tend to show similarities in their calls. And this is known as vocal convergence, which leads me to the next section of my talk, or part 2, to discuss vocal convergence. So first, I would like to tell you what vocal convergence is. So it's specifically a type of learning that results from fine-tuning those spectral and temporal characteristics so that individuals that are forming a bond actually match each other. So animals that form these lifelong pair bonds tend to show those similarities in calls. And specifically, vocal convergence has been observed in birds and several groups of mammals, but it's largely unexplored in other social groups and rodents. And so here, I'm really interested in, before I actually go through all of that, I want to show you an example of what vocal convergence looks like. So here we have budgie calls. So these are 4 spectrograms representing 4 different individuals. And these are their calls before they were put in a group together. And I want you to notice that they all look very different. So some of them are long, some of them are shorter, some of them are at a higher frequency, some of them are lower, the bandwidth is different, right? But overall, you can see that there's a difference between individuals. And here are their calls 4 weeks later. And you can see that each of these 4 individuals are sounding a little more similar, or they look a lot more similar, actually. And then 8 weeks later, they're almost indistinguishable from one another. And this is what vocal convergence really means. Okay. So what we know from vocal convergence literature is that individuals must have this specialized vocal center for learning. And that vocal center is used to modify those spectral and temporal characteristics to match the individual in a pair. And in mice, they actually have similar vocal centers to vocal learning birds. So it is reasonable to hypothesize that they're also able to converge their vocalizations to their mate. So generally, call production in rodents is considered to be innate, as mice deafened at birth still produce vocalizations, but those vocalizations sound different, meaning that they actually do rely on auditory feedback to regulate what their vocalizations look like. So California mice are a compelling system for assessing vocal convergence in a rodent, because remember that they form these long-term social bonds, right? They're monogamous and they're really vocal. So let's see. So for my third aim, I was looking at the effects of pair bond vocal similarities. And what I expected that in California mouse, if there's vocal convergence, then the male and the female in a pair would have more similar call characteristics than 2 randomly assigned individuals. I also expected the pairs that are closer together would sound more similar than pairs that are further apart. And the reason why I expected this was in California mice, there's female bias dispersal, and males that have adjacent territories are related to one another. So I was wondering if there's some correlation between that. And the way that I was able to assess vocal convergence is I looked at my data set that I collected from my AIM-1, so all those vocalizations recorded at the nest site. And specifically, I was able to assign vocalizations to known males and females for a total of 6 pairs. And I looked at differences between pairs and within pairs, specifically based on the 1, 2, and 3 SVs. And the way that I analyzed these calls is I measured 7 different variables at each call. I measured the duration of the call, the bandwidth, or the number of frequencies it's passing through, and then 5 frequency variables at different points of the call. And what I found is that there are differences between pairs in frequency, duration, and bandwidth. And I'm only showing you 2 spectrograms here. Here's one of the frequency variables, and here's duration. And you can see that pair 1 is sounding different from pair 2, or looks different, doesn't sound different. All right. I also found the frequency and duration differences are smaller within a pair. So when I compared those frequency and maximum amplitude differences, I looked at those within a pair and then 2 randomly assigned individuals, and you can see that those differences are smaller, meaning that these animals are sounding more similar to one another. So their spectral characteristics are more similar. And lastly, I found that there's a trend for geographic distance and frequency similarity between pairs. So on the y-axis is the difference in frequency, it's my new term. And on the x-axis, it's the actual distance in meters between pairs. And what I found is that pairs that are closer together are more different than pairs that are further apart, which is opposite of what I expected. So in summary, based on our results, or my results, I found that frequency, duration, and bandwidth are different among pairs, and that pairs sound more similar to one another than expected by chance. And pairs that are closer together are more different in their call characteristics than pairs that are further apart. So the basic functional significance for evolution of vocal convergence is to rapidly identify members of a pair. And so it also helps to coordinate behaviors and strengthen the social bond. So in nature, these social animals, they often separate to forage and defend their essential resources. And they rely on vocal communication to identify members of a pair across a distance, as well as in a noisy environment. So it's important to really consider the actual communication and how that actually will be beneficial for a pair if they are communicating and being able to recognize each other very quickly. So my research adds another taxonomic group that's capable of vocal learning, and there's evidence that vocal convergence is beneficial for both the sender and the receiver. All right, and this leads me to the last part of my talk, which is anthropogenic noise. So I've talked about all of these different natural sounds, but there's also anthropogenic noise, or human-induced sounds. So anthropogenic noise is a pervasive global pollutant that emanates from human infrastructure and activities, such as transportation, recreation, construction, and resource extraction. And natural sounds are present, but they have been around for a really long period of time, and animals were able to adapt to it, whereas anthropogenic noise is more novel and it's constantly changing. So it's really interesting to observe what the effects of anthropogenic noise are on behavior. So one of the negative effects is that noise can actually mask signals, which prevent successful propagation, detection, and interpretation of the actual signal. And for example, I'm showing you a spectrogram in A that represents a red-winged blackbird, and this is their typical call, and B represents traffic noise. And what you see here is that some parts of this call is overlapping with that of the anthropogenic noise. So, which makes it difficult to tease apart for the receiver to tease apart relevant and irrelevant information. And the message is either altered or it can be completely lost in an environment. But anthropogenic noise can also induce anti-predator behavior, such as flight, avoidance, and vigilance, which leads to a shift in allocation of time and energy spent towards being more vigilant as opposed to foraging. And so in this particular pronghorn example, what we see is that animals that are closer to the road spend less time foraging and more time being vigilant as opposed to those that are further apart. So anthropogenic noise and our knowledge on it is really biased towards audible noise, and mainly the literature is focused on birds and bird vocalizations and behaviors. But anthropogenic noise spans across the frequency ranges. So it goes all the way up to the ultrasonic range. And it's really interesting to see how does this broadband, so both audible and ultrasonic anthropogenic noise affects behaviors of nocturnal animals that are communicating in this ultrasonic range. And for my last aim, I'm looking at the effects of anthropogenic noise on activity, foraging, and vocalizations. And what I expected is that anthropogenic noise would decrease activity, foraging, and vocalization production in my sites, and that it would also alter vocal characteristics. So to study this, I specifically looked at deer mice. So I'm actually the deer mice are the same genus as the California mice, but I was able to study them in the mountains of North Carolina. And the reason why they were a great system is really because they're widely distributed throughout North America, and they're a native species, making my anthropogenic noise results relevant across a larger geographic range. But while I was there, I was also able to study or observe behaviors of the woodland jumping mouse too. So what I was able to do is use the same technology that I applied for my previous aims to this aim too. So the same technology allowed me to record these behaviors. But I want to play a short video clip for you. You can see what woodland jumping mice look like. So there are a little popcorn in there. Yeah, there's an interaction that's occurring between these two individuals. But what's really great about this video footage is that I was able to extract a lot of information from it. So I was able to determine the latency for a particular animal to come to the study area, how much time they were spending there, and how much time they were spending foraging. And also it allowed me to differentiate between the species. So you can see that woodland jumping mice are hopping and they're moving vertically in space, and they're easier to distinguish from deer mice, which scurry across the forest floor. So the general setup for my anthropogenic noise experiment is I identified mice in my study area to actually study. So specifically, I looked for resident deer mice, and at the same time, I recorded behaviors from woodland jumping mice. And what I was able to do is record time at that particular area, foraging activity, and number and type of vocalizations produced. And I recorded this for 3 consecutive nights without introducing any noise at all. And then I randomly assigned that site to receive either anthropogenic noise from a road generator or familiar noise from a running creek. And I broadcasted this noise for 3 more nights while recording all of these behaviors still. So I had 6 nights of data, okay? The first 3 nights did not have noise introduced to it, and the second set of 3 nights had noise. And what I was looking at is the difference that we observed between the noise and non-noise days or nights. And what I found is, I want to note that I only put radio collars on deer mice. I did not put radio collars on woodland jumping mice. And this is only for the information pertaining to the radio collared mice. And I found that deer mice spend less time at the study site in the presence of anthropogenic noise. So this is where I was talking about the difference. So the difference between the broadcasting and the non-broadcasting nights, the deer mice were spending far less time there as opposed to the familiar noise. And I also found that deer mice and woodland jumping mice take longer to start foraging in the presence of anthropogenic noise. So again, here's that difference. And if the difference is at 0, meaning that there's no difference in broadcasting, but they're taking longer to actually start foraging. And the one other thing that was really interesting is that it seemed that deer mice were more sensitive to noise than woodland jumping mice, because the first mouse to appear in the presence of anthropogenic noise while it's being broadcasted is a woodland jumping mouse, whereas at sites that did not have the broadcasting, the first mouse to appear was more likely to be a deer mouse. And I also found that deer mice and woodland jumping mice spend less time foraging in the presence of anthropogenic noise. So again, there's this negative number is because I'm looking at that difference. So they're spending less time foraging. And lastly, what I looked at is the number of total seeds that they consumed. And what's interesting is that I did not find a difference in the number of total seeds consumed between the anthropogenic and the familiar noise sites. But I did find the difference in the number of shells from those seeds that were left over. So I'm going to just walk you through this, is that the first 3 nights, there's no broadcasting, and the black bars represent that there are greater than 10 shells that are left over. So at the familiar noise sites, when I start introducing the actual noise, we still see that there's basically, there's no change in the number of shells that are left over. Whereas when we start looking at the anthropogenic noise sites for the first 3 nights, we see the same pattern as observed here. But once the anthropogenic noise is actually broadcasted, we see a total shift that they're leaving no shells behind. So the white bars represent no shells, meaning that they're just collecting all of the seeds and they're running off with it instead of taking their time to consume it at the site. And finally, I found that both anthropogenic and familiar sound actually decreased the number of vocalizations that were produced by deer mice and woodland jumping mice. So broadcasting of any type of noise altered their calls. So I recorded quite a lot of calls before broadcasting, but very few calls during the broadcasting nights. And this is just an example of what a deer mouse vocalization looks like, and 2 examples of woodland jumping mouse vocalizations. So overall, I found that there's a direct impact on individual responses of nocturnal animals, in which we see specifically that mice spend less time in the presence of noise versus no noise. And I also found that there's a negative impact on multiple aspects of animal behavior. So it alters their activity, their foraging, and their vocalization production. So rodents are an important component of an ecosystem, and my research suggests that anthropogenic noise negatively influences their behaviors. And finally, what was really interesting is that there are species-specific responses to the same stimulus. And in my case, deer mice seem to be more sensitive than woodland jumping mice. And so this is important to take into consideration when implementing conservation efforts to mitigate the negative effects of anthropogenic noise. So my research really highlights the importance of assessing the effects of anthropogenic noise across the frequency range and looking at multiple species within the same community. So mice do not like noise. So my overall conclusion is that through different mechanisms is that both external and internal stimuli are actually influencing location preference, and vocalization behaviors. And animal behavior, we have to really think about what it means. I actually want to backtrack a little bit, and I want to note that my environmental stimulus, which is the anthropogenic noise, was more of a negative stimulus, so it decreased the behaviors of location preference and those vocalization production, whereas my physiological stimulus increased it generally, depending on the location. So animal behavior is really three-dimensional. And so it's not just about communication, but also this is influenced by where they are, who they are, and what the social interactions are at any particular space. And all of this influences social behaviors and communication and location preferences. Which leads me to my future work. So generally, I'm just gonna highlight a few of these things, but for all of my aims, I feel like I ended up with more questions than I did with answers. I think that's what's supposed to happen. But what are the actual costs and benefits associated with altered behaviors? And this is important when we consider all of the aims, whether it's physiological stimulus or the environmental stimulus. And then specifically when we're looking at testosterone pulses, I'm really interested to see if pairs are actually coordinating their behaviors and how they're coordinating these behaviors. And then for the vocal convergence, this one is really fascinating because who is converging? When are they converging? So there's a lot of questions that we still need to answer. And then finally, for anthropogenic noise, it will be really interesting to see how other members of the community are responding to the influence of the noise. And then again, why is there a species specific response? So why do we see these responses between the species? And with that, I am happy to take any questions.
Well, Rada, thank you for sharing. This will happen next. We'll have about 10 minutes for the general public to ask Rada questions. After that, the committee members will have a closed section to ask more advanced questions. So here.
That's great. Yes.
So when you were describing the testosterone pulse at the beginning, it seemed like they were relatively short duration. But
Yes.
you injected the mice for 3 different days.
Yes.
Why is that a testosterone pulse?
So I was really interested in the long-term effects of testosterone pulses. So I was interested in how testosterone pulses, because testosterone pulses can have short-term effects, but they can also have long-term effects on behavior. And the long-term effect, one way we can measure that is via condition place preference. Okay. Yes.
So I'm really interested to see if you found any impacts on the pups specifically when the parents stay longer in the nest. And like humans, yeah, that's really good.
So can you hear the questions? Okay, so the question is, can you hear me? Okay, the question is if I found any effects of the parents staying longer at the nest on pups, and I haven't. That has not been tested because that's definitely something that we can look at in the future, and that's one of those costs and benefits of altered allocation of time and energy. So I don't know, but I would expect that to some extent, there would be a positive effect of pup presence, because in this particular species and mention is that male presence is essential for pup survival. So there is a study that was conducted where they removed the father from the nest site, and the survival rate drastically decreased of the pups. Okay, Elizabeth.
So where you found that the testosterone pulses in males affecting females, do you think that influenced pheromonal communication?
So where I found that testosterone injections influenced male behaviors, but then in turn influenced the female behaviors, could there be some type of pheromonal So I don't know. I mean, it potentially could change, but I'm not entirely sure. Yes.
So, I don't, you may have mentioned this, but I didn't catch it. Were you experimenting on the same pairs for both aims for testosterone pulse, the border, the boundary, and the nest?
No, and they were done on a totally different population set. Oh, sorry. Was I experimenting on the same pairs at the nest site that I experimented at the territory site? Okay. No, I did not. So the nest site, specifically, that study was done from January to December of 2015, and the territory was done in 2017 and 2018. So it was totally different groups of animals. Yes. Yes.
Why do you think the deer mice are more sensitive? What do you think? You had to guess or hypothesize the difference between the two kinds of mice with respect to the anthropogenic noise. Because the deer mice have bigger ears or smaller?
They do, actually. So the question is, why do I think there's a difference in behavioral response between deer mice and woodland jumping mice? And there's two hypotheses that I have for this. Number one is that there is a morphological difference in their ears. So deer mice have those satellite dishes for ears, right? So obviously external sounds are really important to them, whereas woodland jumping mice have much smaller ears. So that's number one. But number two, Bettina's laughing, I'm sure, at this, but I've actually observed that there are differences in their behavior and specifically boldness. So in my last season testing, while I was conducting this experiment, I actually did I have anecdotal evidence to support that woodland jumping mice are more bold than deer mice. So there's these behavioral differences.
That might relate to some kind of hormones that they produce?
Yes. Okay.
Do you have some foraging data to support this?
Do I have foraging data for boldness?
Yeah. Is there any difference in response between the jumping mice and the deer mice?
In foraging. So is there a difference in response from deer mice and woodland jumping mice in their foraging? And so specifically, if you're asking about boldness, I was not assessing that in respect to boldness. The way that I was assessing boldness is I looked at some of the papers conducted on deer mice where they were assessing boldness in deer mice. And one way they were doing this is they were looking at a latency to exit a particular area. And in my case, what I did is I opened the trap in the field and I measured the amount of time that it took the individual to exit the trap in the presence of a potential predator, which in my case was me. And so not only did I measure the amount of time that it took him to get out of the trap, but I also measured how did they go out of the trap? Did they walk out slowly or were they jumping out? But again, I mean, this wasn't a full experiment, so this is just preliminary data that I have.
With respect to the foraging data itself, with respect to the husks and things like that, we have information, species-specific foraging behavior that is supporting one species being more vigilant and other species being more bold.
So in respects to foraging data, or specifically husks, if there's species-specific differences, and I don't have this information as of right now, because it was difficult from the thermal imaging information to tell whether one species is leaving husks and the other one is not, because I'm not physically present there. There's not a camera in there in the actual foraging tray to show me that. So, and I did not look at the foraging data over the course of at the video data over the course of the entire night. So how many times did they actually approach or go into the tray and how much time did they spend each time that they spent in the tray. So I did not look at that data specifically. I looked for the husks, specifically what I was able to do is when I set up the seeds right before sunset, I came back the following morning and then I looked at what was left over.
okay? So it just gives me that within that one night, okay, how much of the seeds were left over. And so I don't have species-specific responses to that.
I was just gonna ask if you were pretty satisfied with like your sample size, because I know sometimes it's kind of hard to track down the mice.
Yeah, so was I satisfied with my sample size? Yes, I think actually my sample size is good and it's definitely adequate, and especially from, you know, an ecologist standpoint and looking at wild animals, yes, I would think that it's.
I have a non-ecologist question. Did any of the mice like kind of got used to you and felt like they would just approach you? Did that happen in the field?
Okay. So did any of the mice get used to me or approach me in the field? So did was my presence influencing their behaviors? And I would say no. These are wild animals and they're not particularly, I'm also not there for that. I'm not with them all the time. And I want to note that especially my California mouse study, this was done on a really, really massive grid. And I was only able to trap specific sections at a time. So when I would come back to any one mouse, again, it probably would be days or sometimes, you know, a week in between. So yeah, I highly doubt that. Yeah.
Your testosterone pulsing experiment, was it easy to catch the same mouse 3 nights in a row?
So for the testosterone pulse experiment, was it easy to catch the same mouse for 3 nights in a row? Some mice are what we consider trap happy, so I was able to catch them every single night, and other mice were not. So this is part of the reason why I injected the mice over a 5-day period, is so that it gave me also some wiggle room to catch the mice and look at their behaviors afterwards. But yeah, so there's just differences in if they're, and there's also some environmental factors that can influence whether the mouse will go into a trap or not. Specifically, there's like really big rainstorms, for example. I personally also did not feel comfortable trapping the mice in those specific conditions, but also they were not coming out at their nest site to try and forage and do all of these things, so.
Well, we probably should cut off and proceed as planned. So again, thank you everybody for coming. Let's see.
Staying here.
We have the conference room.
We have this room reserved. I will cut off this live stream. Back.